Hello, everyone. There we go. Got that turned off. Impromptu Friday Q&A. See how many people we get on here in the next few minutes and see how a Friday night goes. Well, that's strange. You got no one watching and then one like. Now it's got 4. What's up, Corey? Welcome to the chat. Hope everything is going well. We'll let some people fill up in here, and then we'll get some questions answered, huh? What's up, Killer? Well, what's up from East Tennessee, Jax, Florida? Hope everybody is doing well tonight. It has been a very good day at Mossy Creek, over at the Foundry, as we call it. What's up, Enzo? Northern England. How close to the border are you? I am obsessed with everything Scots-Irish. I guess that'd be Reavers at that point, though. All right. Well, it has been a beautiful day in East Tennessee. Almost basically T-shirt weather. Just near York. Okay, yeah, so you're not far from where my family's from. And we did a lot of stuff today. Got a lot of things done. Got a lot of videos worked on. Sydney, what's up? Nice, John. Very nice. Heretics of Dune. Oh, man. I'm just rereading Dune. Actually, it is in my camera bag where I keep my computer and everything. And this is the copy I've had since high school. In fact, I have reread it so many times that there are pages that will fall out of it if I'm not careful. It's a good one to listen to. Hello from Sydney. Well, what's up, Santiago? Hello from East Tennessee. Andrew, what should I watch next on Netflix or Amazon Prime? No, Jesse. Oh, my gosh. Okay, so you finished The Expanse. That is the one thing I would suggest. If you want lighthearted comedy, I've been watching The Community on Netflix. If you want sci-fi, let's see. I think you said you've already watched Altered Carbon. Go watch the movie Prospect. It's on Amazon Prime. The movie Prospect. That is one of my all-time favorite movies. Oh, what's up, Angel Hills? Matt. What's up, Yag? Ahoy. Hello, Alyssa. And Magnetic Resonance says hello from the ether. Well, hello from the void, you in the ether. Is there a discount if you buy all available spore syringes? So my liquid culture syringes are not spore syringes, but rather tissue clones in liquid. If you purchase them all in bulk, there is a discount when you buy a bulk pack. It knocks about $5 off a syringe. So by the time you get 9 syringes, you're talking like $45 off. If you buy all the bulk packs, and you're saving about $45 per bulk pack. So, that's a pretty significant discount there. I'll have to see. I don't know how to set up if, you order each one. I'll just talk to my web admin and see if there's a way to do that. But I know the discount's already built into the price. Let's see. Howdy from California. I didn't realize people in California said howdy. Hello from the mycelial underground. I like that. Hello from the void. I hope that helps, Alyssa. Yeah. Well, as I was saying, today was a beautiful day here. T-shirt weather. We ended up getting a lot of cool stuff done. I got some videos planned out, some videos filmed, I guess you can't say filmed anymore, can you? But got some cool stuff edited. So got a couple more videos coming your all's way. Trialing out some new strains, got some new breeding projects going. The employees today Ended up getting a lot done, grow room cleaned, restocked, and got a lot of things rearranged. Got some new substrate trials on the way, so be pretty slick stuff, guys. I'm hoping to have a lot of really cool things for you all. Oh, man. I've got a video coming out, a couple videos from now. It's going to really blow some people's minds, I think. It's going to be awesome. Not the video itself, because I'm a poor videographer, but California has country living too. We all don't live in LA and San Fran. That's fair. That's a good point to point out to me. I do tend to only think of you guys as being coastline, I guess. but there is a lot of country there past that. What's the best bulk grain method in your opinion? Bulk grain, are you talking about like making grain spawn? If that's the case, I'm thinking that probably the best way to definitely for grain spawn. The best bulk method, in my opinion, is the one that I've got listed there. It's the soak method. I do the soak method, throw in a few pellets to soak up some of the moisture, cook it for 2.5 hours. I prefer oats over any other type of grain that I have used, mainly because they can take abuse. So that's the biggest one there for me. Any psilocybin cubensis spores, as in, do I offer any? No, I don't. I don't offer anything like that. If you're wanting that kind of stuff, you've got Sporeworks and Earth Tongue and I don't know, a billion other suppliers for that kind of stuff. Have you ever tried to grow Pleurotus nebrodensis? Yes, I did try to grow Pleurotus nebrodensis, and I did get it to grow. It was almost indistinguishable from a king. I mean, there was a little difference, but that was years ago, like probably going on 2010. And it didn't yield that well, so I just never have gone back to it. I've been soaking rye 24 hours in buckets, but having to boil and dry the grains. Why are you shouldn't have to boil them. Why are you boiling them after you soak them? You should be able to just soak and, I don't know, throw them in a sink and let them drain, and then throw them in a bag with a little bit of like sawdust pellets and soy pellets in there to soak up some of the water, and you should be good to go. I've got a couple videos on grain making. I'd go check that out. Are you all still making the 12-pound bags? We are making 12-pound bags. That we actually have some. When we switch over, we've got a new mix that we're using called the goat mix. When we use it, it's actually more of like a 15-pound bag. Bagging and inoculating old school. Can't for the new building. California permits, baby, here soon. Only took 6 months for permits. 6 months. Oh my gosh. Oh my gosh. That just seems like so much time. I'm really thankful for Tennessee and their low regulations for sure. Can you tell me the best size for the stir bar? About 1 inch. I don't remember what that is in metric. And also, what's the method to select the best culture? Take spore, grow them, and see if it colonizes fast and give good fruit, and then expand it. So you're wanting to go from spores, spores to agar, transfer out from agar to. You can even just do a needle biopsy of whichever one is growing the best. Usually when you select for the biggest colonies on a spore streak dish, those are the most aggressive strains. So you can just needle biopsy, go straight to liquid culture, and there you go. You can do liquid culture. And then take spore, grow them, and see if it colonizes fast, give good fruit, and then expand it. Yeah, you just test out a bunch of strains and then expand those and see which ones give the best yields for you. But typically, the most aggressive strains are going to be what you're wanting anyways, just to outcompete contaminants. Greetings, Andrew. Hello from Northern California. I appreciate what you do, one spore at a time. Heck yeah, Farmer Mac. Thank you. Forget, you say you can expect 1 pound per square foot. What's minimum and max values per square foot? Did it come out to 1 pound per square foot? In our room, it was at 11 by 19. 11 by 19. It's 209 square feet. So no, it definitely wouldn't be square feet. You can expect more than that. And then we multiplied that by 8 feet for our cubic feet. So our cubic feet were 1,672 cubic feet. And we were pulling in an average of about 600 pounds a week for our yearly average. What's minimum and max values per square foot for fruits? For square fruits, so 209 square feet divided by about 600 pounds on average. I think I did that backwards. 600 divided by 209 was what? 2.87 average pounds per square foot. Minimum and max, I'm not sure on. That's going to be strain dependent largely. If you've got some really good strains, I mean, you can bump that up to 3 or 4 pounds. You know, we grow a lot of light strains because we sell by volume. So it ends up pulling our numbers down by weights. But if you're growing really heavy strains, you can oftentimes get more. I mean, you can bump it up 3 or 4 pounds per square foot per week. Have you ever looked into growing Cordyceps sinensis? I've really wanted to learn to actually breed the ghost moths and inoculate the larvae. So, Cal, I have not, but we are setting up a farm, a militaris farm. So we're doing Cordyceps militaris, but I have not looked into actually doing sinensis. So, sorry, guys. Give me one second. Let me mute this Discord. Mute server for an hour. There we go. All right. And with the Cordyceps militaris, we're going to go with a bug farm. So we got the larvae into yesterday, got them into their new little farming setup today, and then the plan is to raise out these larvae for Cordyceps production. I've decided to go insect grown instead of the vegetarian grown militaris, and I haven't looked into I haven't looked into ghost moths as that or silkworms or anything like that, but we're using the superworms for the militaris. Thomas Page says, can't stay, but I'll catch this later. Thanks for what you do, Andrew. Absolutely, Thomas. Thank you for stopping by and saying hello. From what I've researched, no one has figured out how to commercially grow them on the larva. People are growing on rice, but no one has actually grown them on the larva. So, yeah, from my understanding, people are growing the mycelium on rice, and I don't actually know that anyone is actually growing the mycelium out fully, because from my understanding, sinensis is actually an amalgam of different fungal species, and I'm not entirely sure on this because sinensis is not something that I'm like really big on, but militaris is oftentimes grown in like a mixture of rice and soy. So Seiko says, love your hair. Thank you very much. Appreciate that. It's a little flat today and a little I don't know, it's a little stringy, but thank you very much. People are still harvesting in the mountains of Bhutan and Tibet. Yeah, that's my understanding too, is that it's a wild one. I don't know. Somebody's going to crack it eventually. I just don't know when. But the militaris is what I'm going with right now. You're very welcome, Yggdrasil. Any tips for growing chaga indoors? So you're never going to get the fruits off the chaga, is my understanding. Most people will just do that in rice culture, same as what you do with the sinensis. From Hornsey, England, you've taught me so much. I will eventually message you back everything respectively, but waiting to move home and set up the setup. So things are waiting. Can't wait for my home. Heck yeah, Daniel. That's awesome. Paul says, yo, loving the lives, man. Where'd you get the sawdust and soy you mentioned in your last video for wet grain? I'm a woodworker and have lots of sawdust, or should I use pellets? So as long as the sawdust is pretty dry and will soak up water, I think it'll work just fine. You want it to be hardwood, not pine. I think you can use fir. It's going to be fine. I use straw pellets now because of the goat mix. I've always got straw pellets on hand. So you've got straw pellets, soybean hull pellets, sawdust pellets, and you can get those typically at farmers co-ops. If you have a problem getting those, you can talk to Seth Fisher at Mushroom Media Online. He sells a lot of substrates by the pallet and even smaller. So now, I mean, you'll save money if you buy by the pallet in bulk, but yeah. Do you sell substrates or growing mediums? I don't typically. I will to people here local to me, but I don't sell it and distribute that. Mike Tyson does a lot of that. I know. I think Main Cap and Stems does that with certain things. Maybe it's just grain. I'm not sure, but I know there are people who do it. But I don't have the manpower right now set up and the space to just cook substrate and sell it. We're tripling our fruiting capacity in the warehouse. Ben and Bill have been building the 2 new grow rooms we've got coming in. And when we have that, we're leaning really hard into our breeding program. So I'm just not going to have the manpower to do like bulk substrates for people. But there's definitely stuff like I can do it local, but I'm just not shipping it. What do you use to keep your fruiting chamber in proper humidity levels? And is it possible to set such a thing up indoors rather than a farm industrial location? Yeah, absolutely, Armin. I use House of Hydro's sonic humidifiers, and they have a whole kit that comes with a humidistat that you program and a little sensor to program where you want your relative humidity to be. Super easy, super simple. Use code MOSSY when you do it, and it'll save you $1 off your order. But more importantly, you'll get like a full set of replacement discs, and that's like a $50 value. And it does give me a 10% kickback. But there's other things. There's centrifugal humidifiers and things like that to work. Those are much less home friendly than the Sonic Fog. Ready-made, bagged up, ready to poke type stuff. No, I don't have any of that, but Northspore sells boomer bags, magnetic resonance. Northspore sells boomer bags. Mike Tyson sells horse manure bags. There's lots of people doing it. Matt Louder, bugs, cool. Cordyceps, yes. Crickets work well. Okay, I haven't seen people working with crickets for the Cordyceps militaris, but I know the superworms are supposed to be really good for it. Do we eat the bug from sinensis or just eat the mushroom on top? I don't know on sinensis. I've seen people eat the bug and everything with the cordyceps. I mean, I'm using superworms, and I know people actually use that as a food source. Like, people eat those. But we're going to trial a few different types of bugs out and see what works best. Thank you for the time you put on the share content and information, dude. Oh, you're very welcome, Armand. Did you study mycology? You're a effing well of knowledge. I did not study mycology. No. I'm starting, but I'm scared and $3,000 in. So, Stephen, I went to college for less than a year and then dropped out. So if that tells you anything, I was going in for myrmecology, which is the study of ants. I'm probably mispronouncing that. And then I went into microbiology, but, then I started growing mushrooms while I was in college and found out that I didn't need a bunch of the cool thing about mushrooms is that it's a fringe science still. And so amateurs can make huge leaps and bounds in this and contribute very heavily to the way things are going to be without a degree. So, and I found out that I didn't need student loans to do that. And that's why I did that. In about a month, I want to get the bagger. What's the best way to go about that? Just order it off the website. That's the easiest way. In fact, we're working on doing a much larger logistics deal, selling just straight, like selling it and having some help getting logistics in, so we're saving people a lot of money. John says, damn, lagging a lot, but I shut down inoculating for the chat. Gotta have priorities. I'm sorry, John, man. I didn't mean to stop your work. What is the best substrate mix for growing bear's head to? So the best substrate mix I have found for bear's head is 50-50 mix of pine pellets and soybean hull pellets. That we were getting 4 or 5 pound first flushes off of 12-pound blocks. Did you say Hericiums do well on Douglas fir? From my understanding, they do. I've not ever actually grown them. It's just what I've heard from what the growers out west. Thanks. The Mike Tyson with no, so not the Mike Tyson. It's spelled MYC as in mycelium Tyson, and he's on Instagram, and I don't know where else he is, but he sells all over the place. His logo is literally one of his stickers is a mushroom with looks like Mike Tyson's face with boxing gloves. So, hey man, what temp do you incubate grain spawn at? Just room temperature. Right now it's sitting around 65 degrees. Just in my lab is I think it's at 65. Tito says, hi, sir. You look very nice. Thank you. I am your number one fan here in the Philippines. More power and God bless. Well, thank you, Tito. Appreciate that. Sir, please give me a simple working liquid culture recipe because I'm just new to this field. So a simple working liquid culture recipe, I mean, 10 grams malt extract, 10 grams corn dextrose, 500 milliliters of water, cook at 15 psi for 30 to 40 minutes. There you go. Glad to hear you're growing. Oh yeah, Matt. We're going to be hitting Cordyceps hard. I've had a lot of people requesting that for the YouTube channel, so I'm going to set up a couple of different ways of doing it. We are going to grow some vegetarian wise, and I'd really like to get some sent off to a lab because I have an instinct that the bug grown is going to be much more nutritious than the vegetarian grown. His video in his fruiting room is amazing. Check it out. On Mike Tyson's or mine. I haven't seen Mike Tyson's fruiting room yet. John Grady, should I be restarting my cultures from spore after a certain amount of generations, or do I grow out the mic on sub low in sugar to prevent the enzyme blindness? So, John, from what I've been hearing, enzyme blindness has actually helped out a lot in liquid culture by the stirring of by the shredding of the mycelium by the stir bar. Jesse Campbell was telling me he read that in Radical Mycology. Somewhere I must have missed it. And how that keeps it going with, and keeps it. I guess it's the disturbance that helps with that. But I don't know. No, I wouldn't. So what? If you're worried about enzyme blindness, just make up some wood agar, and that's basically you do like 10 grams of or not 10 grams. It's like 5 to 8 grams of hardwood fuel pellets. 10 grams of agar, 500 milliliters water, cook and sterilize, pour into plates, and then there you go. Just grow it out on there and grow it on either one side or from the center, and then select for after it's crossed the dish. And there you go. It's just been eating wood that whole time. Armin, would a plastic liner in the room be sufficient to keep in humidity and not cause mold in a building? So I'm not sure what you mean exactly, but I'll tell you that my grow rooms are they're in a wooden building, block and wood. And all we did was we did a 2x4 structure, and then just did plastic around both sides. So we did an interior wall and an exterior wall, and then insulation in between, and works really well. A giant Martha, essentially. Yeah, I mean, that's... Yeah, as long as you're sucking air out, and you're not you don't have a lot of air escaping into your room, you should be fine. But you want to make sure you're definitely ducting that air outside, because you don't want spores in your house anyways. You're welcome, Magnetic. Campbell Walker, good day, Mossbacks. Well, good day, Campbell. Are you able to suggest a size in terms of voltage for a sauna steamer to be used for a bulk pasteurizing unit? The 9 kilowatt, our sterilizer now, our boiler is a 9 kilowatt boiler. So, you're not going to do a full ton of substrate with that. That would be about the most I can suggest for you. Ben Erickson, one of my business partners, is the person to really ask that question to. And I don't even know what kind of answer he'd be able to give you. It'd probably be, something similar. Armin says, technically you did study mycology, but without college. I guess that's true. Maybe let's call it, I'm not a mycologist. I guess I am engaged in research. Man, I wish Adam Bolton was here now, because maybe he'd be able to parse this out for me. Maybe I'm more of a mycological engineer. Something like that. Maybe that'll work. Or a mycological miner, which is why my gamer tag is Myco Miner. But maybe something like that. I don't know. Matt Lauder says, love you, bro. Got to get back to work. Oh, shoot, man. I hope I got to you before you left, but take it easy, man. Get back to work. Daniel says, have you ever set up a worm farm in an IBC from your substrate and sold your mushroom compost? I have not set up I've got a worm farm in my kitchen where we put our kitchen scraps and coffee scraps and stuff. I am going to be moving it over to the foundry, which is actually that direction, not that direction. And I'm going to be attempting to grow, to use spent substrate in there, but I don't know how well that's going to work. I'm afraid that the oyster mushroom substrate, the oyster mushroom mycelium, may kill and eat the worms. I will say that the superworms that we have, we broke up spent blocks out of the grow room, put them into a 10-gallon aquarium, and then threw 1,000 superworms in there. And darkling beetle larvae, which is what that is, eat a lot of them eat fungus just in nature. We've got a few carrots in there mixed in as well, but largely I'm hoping that they'll eat the spent blocks. I saw that William Pitty Brown did something similar, and I think that that's going to work out really well. And I am eventually, like, my worm bin probably actually has enough worms in it now that I can take a bunch out. And I use the worm wee, the liquid drippings from the worm bin, to feed my fish tanks. So I've got a fish tank set up in the kitchen where I grow a lot of like edible plants like watercress, water spinach, taro. We got duckweed for the tilapia, things like that. And the worm drippings do really well. Like, I've not had to buy any artificial chemicals, fertilizers, anything since getting that worm bin. And that was at one of my former employees, Jason, who's If It's Fish on here. He suggested that, and when I started doing that, man, it just blew my mind at how well it works. Pine fir tree, as in the Christmas tree. I don't really know. I know fir trees are supposed to look evergreen, but yeah, people use fir sawdust all the time. And as far as I know, it's not a pine tree. Fir is it's different species, different family than the pine family. I was talking about your growing room. Okay, I got you, Magnetic. Yeah, well, thank you. I'm not sure if you're talking about the new grow room or the old one at that point, but both of those grow rooms, I'm sad to see the old ones getting taken down, but the new ones, oh my gosh, they're coming together. It's beautiful. I mean, we're just just the amount of grow space I'm gonna have. It blows my mind. I never thought I was gonna get here, so it's kind of cool to see it. You're welcome, Arman. In about a month, I want to get a bagger. What's the best way? Killer, it's just ordering it off the website. I mean, you can literally just order one of our baggers off of our website. We have a bunch in stock, and we're making more all the time. I think we've sold 20 something of them since October of last year. So we've got plenty more being manufactured constantly. Knowledge says, "Yo, what's up, Knowledge? You are a farmer." Well, thank you. Yeah, that is a noble enough title for me. I like it. Jumbo Jet Canada. Gotcha. Are psilocybin mushrooms difficult to grow? Some of them are, and some of them are not, is my understanding. I don't have a lot of experience growing psilocybin or anything like that, so it's hard for me to tell. I've consulted on some, ops that grow it, that are getting ready for, legalization out west. And no, I mean, like, cubensis is not difficult to grow. It's as easy as an oyster mushroom is to grow. But I've heard like some of the stronger types, the wood lovers and things like that, are more difficult. But I don't really have any experience telling you, to tell you. So, let's see. I like that, mycelium engineer. Yeah, see, that's kind of, I like that title, but I still, if you have to go to school to be an engineer, then that's not the best title. Then the mad scientist. Yeah, I mean, I've always kind of liked that title. So the solarium period was the period where prototaxites, ruled the earth, when like the largest structures on the planet would have been like mushrooms in this one tree that was very similar looking. And I had this character for a video I was doing one time that I was calling Dr. Solarian, and I never made that video, but it was a mad scientist video where I was Dr. Solarian from the fungal, the great fungal period. I see a lot of debate about light as a pinning trigger. Are you able to weigh in on this debate? Some argue the fresh air exchange is more important. Restrict this to oyster mushrooms and hericiums. So if I'm restricting this question to oyster mushrooms and hericiums, it's strain dependent. Some strains grow amazingly well pinning off. If they get a little bit of light, they'll just pin like crazy. Some of them, it seems like cool weather is a trigger. I have not been able to figure it out as a species thing. It seems to me to be very strain dependent. So that is actually some of the data that we're wanting to take in the greater archive. But right now all I'm doing is yield data. Oh, shoot. I forgot to bring all those sheets home with me. I was going to plug those in later on tonight. That's okay. By the time we're done with this live stream, it'll be like 9 o'clock. It'll be time for me to take a nightcap and go to bed. So, John, I don't have a good answer for you, other than it's strain dependent. And I know, as far as I know, no one has a good list of those strains to tell you. Christian says, "Hey, Mossy, how about the dog agar? I've never done dog agar, and I can't see a good reason as to why I would do it. Mushrooms don't really eat meat, and I guess dog food is mostly vegetable material, but why, whenever malt extract and, all the other ingredients are so much cheaper than dog food even?" Let's see. Those cycle grow methods are absolutely boss. Cycle grow methods. You're going to tell me what that means, Armin. Have you tried using old blocks for growing other foods? We use old blocks in our garden and rose garden and vegetable garden, and we have some amazing vegetables and flowers. So yeah, I've absolutely used it for that. I haven't tried it directly for growing other food. The insects are. I've grown black soldier fly larvae off of spit and mushroom compost, and this summer I plan on setting up another black soldier fly farm. Armin, yeah, you can ask questions about psilocybin. I'll tell you whatever I know. I don't know much about it, but I'll tell you what I do know. Lance says Azurensis sucks. Okay. I guess I've definitely got no experience with that. I don't know anything about that. I've spent a lot of time on the shroomery, and it looks like cubensis is incredibly easy to grow. Azurensis, I don't know if you mean it sucks to take them or sucks to try to grow them. Yeah, Jumbo, you're very welcome. Have a good weekend too. He's just a few comments behind you guys. Be patient. He's reading them all. Yeah, it takes me a while. Plus, the chat will spend a long time just doing nothing, and then it'll jump up real big. So a lot of times, like as I'm reading, I don't think that that I'm that far behind until all of a sudden it just jumps. So I'll take a little bit more time than I always need to. Santiago, can we grow mushrooms in a bag without a box covering it? I thought that mycelium will grow only from the square of the box, which enables light. Is this true? Santiago knows. If you've watched my videos, you've seen we just fruit right out of the bag. The bags are clear. No boxes required. Mad Max Inc. I love the name. I'm having trouble getting second flush fruits on my lion's mane block. Any tips? On Hericiums, I don't get a lot of second flushes on lion's mane stuff. I get heavy second flushes on comb tooth. But usually for your lion's mane, it's just waiting. Just time and waiting. Fungal fruits, what's up, Dan? Hey, late as always, Spartan blocks. How's that going? I'm out of the loop. Must be close now. Yeah, so I'm about to do a video on that. We just set out the Green Elf Cup blocks and opened them up on the back dock, to start drying. And then we're going to run those through the bandsaw. And once those are through the bandsaw, we'll hopefully have them completely dried out and run them through the planer. The zoning blocks are also about to do the same thing. But I think next time I do like the black lines zoning spalt, instead of having the bag sitting like they normally do, like that, I'm going to lay them back and I'm going to have a much thinner layer. That's going to be much easier to run through a planer. And I'm going to use a bandsaw just to clean up the edges and then just run it through the planer. And then I'll have my sound panels for the studio. Farmer Mac, Andrew, you're one of the best microbial ranchers in the world of fungi. Thank you. I actually like that. Microbial rancher is a good one, too. You can also call me the Swarm Master or Abathur, or I'm just playing. I'm making sci-fi jokes now. Letting the old geekiness show through a little. You're very welcome, Santiago. What are the minerals that mycelium loves, and what are the minerals that mycelium hate? It's going to be entirely species dependent on what they like and what they hate. It's like asking what minerals do plants hate and what minerals do plants love. In fact, it's even harder than that because there is a greater diversity of fungi than there are plants. So, yeah, cubes, just corn and core. I'm trying my hand on lion's mane. Yeah, lion's mane's a good one to grow. It's a good one to sell. I like that one a lot. Oh, what's up, Shaka? Gotcha. What's happening, man? Can you help me use terms correctly? What is a strain, and what do I call mycelium? I've sectored from plates and isolated. Is it an isolate? Okay, so you've asked me like 4 questions there, John. And yeah, I'll absolutely help you parse that out. So hyphae is a single strand, right? That the fungal material, they grow in like a cell grows and then repeats and then repeats and then repeats in a linear fashion. That is a hyphae, is a strand of cells. A collection of strands of cells is called mycelium. And then a strain is it's a little tricky to parse out in real scientific terms, but in working knowledge, a strain is a group of mycelium that all share genetic material, right? They share the same genome, and they share the same set of DNA, and they're going to have repeatable results. So let's say that it's crazy because mycelium, let's just use this whole screen for an example. I've got different types of substrate here, right? This mycelium grows in, this is all one strain, but let's say one of these takes off faster than the others, and it grows across this different type of this new substrate faster than these 3, right? And then I isolate off that, and you can typically see that because it'll take off and it'll start to branch out, and these will kind of grow in slower, and they didn't gobble up as much territory, so they kind of just continue going that way. The one that gobbled up all that material, I would call that more of a substrain than an actual strain. It is an isolation, and an isolation of a single strain would be, to me, a substrain, or a variant, right? So you have different plant variants. Like, let's say I take Kosovo tomato seeds, and I plant them here in Tennessee, and I grow them for 7 years, saving the seeds from the plants that do the best. I now have Kosovo tomato variant, Jefferson County, Tennessee, number 7, right? 7 years. That would be like a substrain, so to speak. You have acclimated that strain to that material. Now, let's say that you take spores, and when you take those spores and you do spore streaks on a dish, and you see all these obviously different colonies, and you isolate that out. That is a new strain because it was derived from spores. And that new strain now has new genetic potential that has been yet untested, right? You're going to test that out. Once you are copying that over and over again, let's say like the new strain that I just released, the BS21, the super scallop, which is a blue snow cross. I selected for that one because of its aggressiveness. When I tested it, it had this crazy scalloped edge, and I codenamed that one Super Scallop, and then ran it through commercial testing now, right? That strain, I've had it for about a year, and I'm just now getting to testing it, just to give you an idea of how long it takes to really churn through strains. And lo and behold, in our commercial test, if you can go look at it on our website and see all the pictures, all of them are scalloped. It's a repeatable result. That is now a strain, and that strain is called Super Scallop. It is a heavy fruiter, a beautiful mushroom, and all share similar phenotypic characteristics. Phenotype is the visual expression of genes. So I hope that answers your question, John. This is the kind of stuff that I love. And I'm really hoping I'm not boring anybody, because, John, I feel like you and I are probably like geeking out about this, but this is my favorite part, like the breeding, the isolation, the seeing the new strains come out. This is stuff that gets me excited. So would adding a small amount of oat water to your LC recipe help train the mycelium on your grain, speeding up inoculation? Manheim, that is one of my tests I'm doing right now, is a grain broth and a wood broth. In fact, I'm wanting to see if I can just do a grain broth. Like, let's say I take my water that I've soaked my oats in. I'm just going to take that water the next time we drain and just do 500 milliliters of that and a couple of times and see if it just takes off, if they don't need much in the way of sugar water at all. Chip says, howdy, Andrew. Thanks for all the great content you rock. Well, thank you. I'm always loving that the positive feedback. By chance, do you have any colonized grain spawn ready to sell? Guess who ran out? And howdy, Mossbacks. So, Chip, I am back ordered. So, no. You want something fast, go to Main Cap and Stem or North Spore. I've got some really cool stuff you can get from me, but it's going to take at least 3 weeks on any given strain. And on some stuff, it's going to take 6 to 8 weeks right now. So I am being hammered hard by grain spawn orders. And I just got in 2 new autoclaves, and I was only able to set up one And I've got to get the third one. I've got to get the electric run for it so I can get the third one so I can do 18 bags a day. But I'm not there yet. I mean, I'm still back ordered on some orders to November and December of last year, if that gives you an idea. That's certain strains. For the most part, everything else is going out well. And I mean, already just having the second autoclave is speeding things up like crazy. I appreciate the time you spend giving us helpful advice. You're very welcome, Magnetic. Thank you for listening. Thank you. One other, any recommendations on best substrate for Cordyceps? So Mad Max, I don't, my guess is bugs are the best substrate for Cordyceps. Especially if you're talking about Militaris, I mean, any others, but I don't know for sure. A lot of people talk about growing on rice and soy and all this stuff. And to me, it's just like, why train a lion to eat tofu? Like it's just not going to work. It's just not going to work as well. Give the lion its actual meat and it's going to perform much better. And I want to do a vegetarian grown and a insect grown Cordyceps militaris. I want to use the best substrate that a lot of these guys, the more hippie types, are using, and see, like, is this vegetarian grown one really nutritional value, like higher nutritional value than the bugs? Because I'm going to guess the bugs are going to be providing a lot better trace minerals, especially when I'm gut loading those bugs with what they eat in nature, which is fungal material. So that kind of stuff. Lance says growing azure incense. One of my best friends live in Westport, Washington. Okay, that's where I hear they come from. They always are better boots on the ground. Gotcha. Makes sense. Hit the thumbs for support. Yeah, thank you, Magnetic. Everybody hit that thumbs up for me, please. That helps me out a lot. And if you haven't subscribed, please do. The subscription helps more than anything. Fungal fruits. Awesome. Can't wait to see these sound panels. Looking forward to it. Me too, man. I'm really hoping they work well. I'm hoping I don't have to like resin cast them or anything. Paul Hannock, from henceforth you shall be known as Myco Jedi Master. I hope I don't get sued for that. From what I understand, Disney's pretty sue happy. John Grady, as a small farm growing oysters and hericium, should I bother with oyster shell or gypsum? I've never done that, man, and I've never heard, like any of the big farms I've listened to, I've not really heard of them doing it. I think What the Fungus does some. I think he does oyster shell, eggshell. I don't know. I don't see why you should bother with it. Maybe try a few bags and see if it bumps up your yield, but my guess is it's not going to do anything. Has anyone figured out how to grow Liberty Cap successfully? Just wondering what you've heard. I don't know on that one. If Liberty Caps are what I'm thinking of, they're the teeny tiny little ones. I've never heard of anyone growing them. Just I've heard of people harvesting them wild. Spore syringes are $12, $15, but what do you think I should charge for my work on LC? Friends starting to ask if they can buy syringes of my LC. Well, I mean, so if you test regularly on agar or on grain, and we're talking about something non-illicit, I assume, because illicit, like once you get into black market stuff, you're talking like super overcharge for everything, I guess, but I don't have a lot of experience with that. If you're talking about like just liquid culture, then I would say anywhere from $20 to $30 is actually really good for well-tested strains that are tested on the regular, and that are novel and new, like wild strains. If you're just getting like liquid culture from someone else and then expanding that, then yeah, $7, $8, whatever, that's fine. And you're not doing a lot of testing. That's cheap enough that, people will take a gamble on that. But if you're wanting to sell quality stuff, then yeah, definitely do high quality testing, always be searching for the best strains, that kind of stuff. So $20 to $30 to me is a pretty reasonable amount. In fact, I've been told by a lot of people that I ought to up my prices somewhere for like my bread strains for like anywhere from $100 to $200 a syringe. I am not going to do that. I have so many new strains coming out and so many strains that are regularly retiring that I don't think that I need to charge that much. I just don't see that. I think it's going to be much better to get a large diversity of strains out to people at a reasonable price. And so I think $20 to $30 is good. Spore syringes, yeah, something like $12, $15 is probably good if you're taking good clean spore prints. Justin says, hey there, can I mix spores together from 2 different mushrooms, but both from the same family? Well, I need to get a new crossbred strain. Yeah, that's the only way to get new strains. And it's not it is a cross, not a hybrid, right? Unless you're getting 2 from 2 different species. And if you're talking about from the same family, that's a little bit different than species, right? But if you're talking about like Pleurotus austriatus, right? The oyster. I know that it will actually cross with Pleurotus austriatus variant columbinus, the blue oyster, because I've done it many times now. Yeah, you take your spore streak, and then where you have your intersections of the 2 spore streaks, whatever you see growing from that, you can sector that out, plate it out, grow it, test it, and bam, you have new strains all day long. And in fact, I am doing videos on not only breeding new mushrooms, but on how I breed. That's a little bit cheaper and faster way to do it. So Farmer Max says, yes, more geek talk. Well, thank you. That's the kind of stuff I get into, man. Like I love talking breeding mushrooms. I think we have not even begun to see what selective breeding can do for mushrooms, and it's going to be bewildering. Like I was talking about that super scallop with the actual scalloped edges being consistent across the strain, blew my mind. We got this new oyster that we're trialing out right now that we're calling the watercolor oyster. The watercolor oyster looks like somebody daubed a little bit of watercolor on, you can see it on my Instagram account. I think it's on the Mossy Creek Mushrooms Instagram account where I'm talking about some new strains, but it looks like somebody just daubed a little bit of watercolor and the watercolor ran over the paper. And it's beautiful strain. It's weird looking. And I did not think it was going to be consistent across the strain. And every single one is doing it. When they come out as pins, they have nothing that looks like it. They look like there's some blue pins, some brown pins. And why the pins come out different colors, I don't know. But they'll come out, and then as they grow and the caps unfurl, they get that watercolor tinge to it. It's weird. And I'm just loving I've barely scratched the surface. And once we get a lot of people breeding new strains and then trading strains and then breeding those together and then doing isolations and seeing recessive traits, we're going to see an explosion of diversity across the mushroom hemisphere, the mushroom world. It's going to be crazy. Would you sterilize your grains in your steam sterilizer, bulk sterilizer? If so, how long would it take? So, yeah, sure. I've been thinking about throwing some grain bags in just for the production blocks. Just throwing them in there and cooking them for 24 hours, like I'm cooking just alongside my substrate, bringing them in and using a grain master to inoculate them, and then using those as my regular standard production, because you can get a good 1-2 generations of pasteurized grain to work out really well. I would not use it as a grain master because by the 3rd or 4th expansion, when I did that, things were getting really dirty and my transfers were not clean, being very clean. So Family Guy line on tofu skit, perfect on the cordyceps, effing hippies, long wheezy cough. Yeah, that line is exactly what I was referencing. I freaking love that show. Futurama was so good. Thank you, Mad Max. I appreciate that, man. Best book or several books for me to purchase so I don't have to ask you so many questions. I burst out laughing when you apologized if it was too deep, boring when you answered my question. For mushroom growing, Radical Mycology by Peter McCoy. Let's just say if you're not a ultra progressive, just ignore that section and just ignore all the political stuff and go right to his technical materials and just, read that. If you've got a good stomach for progressive stuff, then it'll be just the book for you. Unfortunately, there's not really a lot of just good technical manuals without, mushroom philosophy BS that a lot of people have to get. And I love mushroom philosophy. Don't get me wrong. It's just, why doesn't someone just have maybe Ben and I will write. We'll call it the No BS Mushroom Bible or something like that. I don't know. And it'll just be technical information for how to grow, none of our own philosophical BS mixed into it. That way we don't have to, try to infect you guys with our philosophy while at the same time giving you the technical stuff, right? The old bait and switch. Radical Mycology is a good one. Growing Gourmet and Medicinal Mushrooms is a good one. Trad Cotter's got a book that's pretty decent. But I feel like Radical Mycology really blew it away. Who else? again, like I'm rereading Growing Gourmet and Medicinal Mushrooms by Paul Stamets, and a lot of this stuff when I was reading his book, I was like, oh, this is great. And then radical mycology, and I was like, man, Paul Stamets stuff is so out of date. And then went back to Paul Stamets stuff, and a lot of things that I'm doing that I'm like, man, this is so innovative. Paul Stamets is talking about that stuff like back in the 1980s and 90s, and I was like, oh, okay, that's some good stuff. So there's that. So I definitely we see your shadows, Brody. So those two books are really, really good. Like I said, Tread Cotters is pretty good. Mycelium Running is actually a really interesting book for a bunch of low tech stuff. That is very distracting. Well, you need to stop. It's rude. And let's see. Boy, you are being entirely too distracting. Those are the two I would really recommend. The Mushroom Cultivator by Paul Stamets is a good one. Oh man, What the Fungus is supposed to have a book out that's supposed to be pretty good. I've not read that one yet, so I don't know, man. If you get Brian Callows of What the Fungus, let me know how that book goes. But Radical Mycology is like the first book I would recommend because it's so complete that you can forgive it all the political stances that it takes. You like your All-American? I love it. I've got 3 now. I see some generics on Amazon for $200 versus the $1,000 for the American, but of course the cheap one may explode trade-offs. Man, I like the autoclaves that I'm working with now are amazing. I love it. Once you get it dialed in, like what I did, it floats. The electric sterilizers, the PSI will sit there and float back and forth, and you'll sit there and you're going back and forth and back and forth. And the good news is that if you can just full crank it till it hits 15 PSI, and then back it off and bounce back and forth until you find where it maintains at 15 PSI. You can mark that with a white paint marker, and it's much easier to use. You just, bam, go that till 15 PSI, go back to where it's at, and it's all autopilot. And it is beautiful because 18 bags a day is much better than the 6 I was barely able to eke out constantly. So, Lance, for a dumb butt, like AKA me, do you recommend spore prints or syringe? If you're going straight from spores, just buy a spore syringe. You can use it just as easily as a spore print, and they're still hydrated. Assuming that you're talking, what I think you're talking about. Damn, 18 bags a day. Nice to hear how your business is growing. One day, Cetus will get there. Please tell Ben he needs to do some more hard truth videos. You two make us think out of the box. Thanks for everything you do. Have a great night and keep streaming. Absolutely, Chip. Thank you. Keep those good Instagram posts coming too. Oh, and Cetus, I did work out today, just so you know. I definitely did some lifts today. I did the overhead single arm 32 kilo press today. I did 0 cardio today. I was not feeling it. And I do think I'm going to get my bike together so I can start doing more cardio, though. I'm thinking about just biking back and forth to work, but my backpack's not very comfortable for that, so we'll see. Fungal fruits, breeding. Do you think it's possible to breed resilience to blotch, for example, into your strains? Yes, I do. And that was not my initial, like what my first thing to do was. Mostly I was interested in new and novel phenotypes when I started breeding. So like the King Blue is a little bit blotch sensitive. It's usually like the first thing to blotch before anything else is. And it's actually a really good indicator mushroom. I'm actually thinking, how interesting would it be to have mushrooms that are purposely like sensitive to things? Excuse me. Like, what is it? Like you have a canary in the coal mine mushroom, right? Oh, this strain started to blotch today. Time to clean the humidifier. Something like that, little indicator strains. But yeah, no, I think blotch resistance. So there was a time when everybody was growing North spores, Kong. I think it was like the King Kong, King Oyster or whatever. And everybody across the country started having blotch problems at the same time. Like everyone. And then I mentioned maybe it's a strain thing, right? Like maybe that strain, maybe this year we're having a particular type of bacteria that's common across the country. and that the strain may be susceptible to that type of bacteria. Then I had some people attacking me saying that I shouldn't attack Norspore that way without proof, and I was like, I'm not attacking Norspore, I'm attacking the fact that the strain, everyone's growing a monoculture, and as it happens in monocultures, this thing may be the case. So I grew out 3 different king strains. One of them was Kong, and I grew it right in the middle, and then I grew one of my old king strains, and then a variety from who did I get that other variety from? Was it from Aloha? It doesn't really matter. I grew the 3 strains out, and the one in the middle was the Kong variety, and it absolutely blotched all over the place. And then the other variety from Aloha, I think it was, that one just kind of blotched, but not bad. And then my old king strain grew in, and it didn't blotch at all. And I was like, okay, well, that's pretty obvious. There's one single strain that's blotching pretty bad. And I don't think it's a bad strain. The Kong variety was great. And I don't think Norspore was like sending out infected blocks or batches of spawn or anything. I think that what it literally was that everyone was growing a monoculture. That's kind of what led me to going, I think we need a greater diversity of mushrooms, and I think we need breeders breeding in resilience to different things so that we can see where selective breeding can take us, both in yield, but also in disease resistance, aggressiveness, speed, incubation, phenotypes, weird new things. Like, how cool would it be to go to a farmer's market, and instead of seeing like one blue oyster, one gold oyster, whatever, you're seeing like 15, 20 different types of phenotypes. Like all of these oyster mushrooms, they're all oysters, but they look like different species, right? They look like different things, kind of like what we've done to dogs, like the very same thing. So yeah, Dan, I think that we can absolutely breed that in. And the chat just jumped. Give me a second. I've been learning cultivation off YouTube, but now I feel the need to read some book for troubleshooting. Can you recommend some books, please? Okay, yeah, Daniel, I hope that my book recommendations already got you there. So, Cameron Ferguson, what's up, bud? How are the bug inoculations done? Do they ingest it, or do you grind them up, inoculate them as a substrate? So there's a few different methods. I'm going to try the substrate method. There's also people who will like literally take the time to inject every single bug with the cordyceps, like liquid culture. And then there are people who I think do spore dispersals. So I don't know yet. I'm going to be doing a variety of ways and then videoing every way that I do and show you guys which one I think is the most successful. But just look up like cordyceps inoculation on YouTube. You'll find like, especially in Asian countries, they do like actual injection of the insects. A lot of times, not all of them. There's a lot of vegetarian grown ones too. Stamets is good, easy reading. I agree, for sure. I'd buy it. The Mushroom Bible looks like Ben and Andrew need to make the Bible done. I'll talk to Ben about it. We'll start. Maybe when we're done with the grow room build out, we'll start on that. Hope there's many more that see your knowledge because I'm 8 months in and want people to open their eyes to freedom. Oh, thank you, though. I appreciate it, man. Well, love the shadow puppets in the background. Yeah, that was my son. Call of the Little Ones. Yeah, they love to come do that to me, and I'll be seeing that stuff, and they know my ADD kicks in, and I cannot focus on that, on like answering questions when that's going on. Everyone needs to just pester him to start a journal and then construct a formality of substance. I'd be so happy to see this stuff on a production level. Yeah, man, I mean, I don't see why we couldn't just sit down and write a book. It shouldn't take, like, if we did it, took some time off, like Ben and I, and just let out, like I've got Jackie fulfilling orders and stuff. Maybe I just need to take some time off and sit down and write a book. Like no videos, no nothing, just a month of just sitting down and writing a book. I don't know. Chip says, Snicker, how much do you want to bet Brian's interns that with Fungus wrote his book? I don't know. I think that Brian actually took time off. I know when he was about to have a baby, he like sequestered himself into this little like RV or whatever, and then to take the time to write the book. I've never actually read it. And so maybe he wrote it based off questions that his interns asked him. And that would actually be a really good way to write the book. I should do that with my mentorships. The next mentorship that comes in, I'm going to have a list of questions for them. And I'm going to write down all of their questions. And I'm going to build that into the book. That's a good point, Zetas. Things are there. Dark waters is nothing compared to the things people actually experience on a daily basis. Don't know what you mean by that, Daniel. I'm sorry. I must have missed something. John says, selective breeding with mushrooms. Enter the Benny Gesserit. So, yeah, man, I'd love to be Benny Gesserit with people too, but that's frowned upon these days. We're building our first dedicated building for our mushroom biz, 1,000 square feet. Any recommendations on layout or design that you found helpful while you moved into your new space? Sure. So you're going to need more grow room space than incubation space, unless you're doing shiitake, in which case you'll need 10 times the amount of incubation space that you have grow room space. So with that small of an op, stick with fast growing species, fast turnaround, high value stuff like oysters, hericiums. Maybe dabble a little into like chestnuts, black poplars, things like that. That take like 6 weeks, but don't do a lot of those. Definitely focus on doing high speed, like high turnaround, fast turnaround ROI stuff. So like oysters are perfect for that kind of thing. You'll says, "Low, forget cardio, do legs and back." Yeah, I need to do squats. Maybe I'll do some goblet squats tonight before I go to bed. Since you're sitting around doing nothing lately, why don't you do more mead content? So Lance, I'm not sitting around doing nothing, as I'm sure you know, pushing that button. But I could do some more mead content. Actually, it's about time to bust out the Forest Dwellers mead and give you guys, like, I still have that jug sitting down there. We ought to just bust that out, pour it out, and then I'll cook with it. Maybe a cooking video. We'll bust that out and have that going. And I could do more mead content. If people are interested in brewing, I can absolutely brew more. I just figured people would be more interested in mushroom content. And I've been getting a little burned out there this past summer, just with all the sheer amount of work we've been doing and mushroom stuff. I've just been kind of wanting to get away a little bit. But brewing would be a good way to break back into that pretty easy, huh? I got behind a bear's head, started fruiting filter patch. How would you proceed with fruiting? Just let it fruit out the filter patch at that point, man. Like, it's crazy. Like, just let it do it. Yeah, more diversity, like in nature. Yeah, I mean, that's what I want. Like, so the thing about the interesting thing to me about oyster mushrooms in nature is that they don't tend to like out in the wild. They all tend to look fairly similar. I mean, there are differences, but they all tend to look very similar. But then when you bring them into the grow room, they all respond like crazy different. And we're in our Discord server. We have a plant geneticist, and so he's been reading up on fungal genetics for, like, not for me, but like with me, to help us out on these breeding programs. And there is a lot more at play in fungal genetics than any of us guessed, or maybe many of us guessed. But it gets weird. What do you call them? Transferons? Like, I don't know. There's just a lot of like turning genes on and off in mushrooms. So it's just, it gets really weird. Like, how much of that expression is genetically there and not there, or how much of it's just being turned on and not turned on, right? I don't know. On average, how many mushroom blocks is needed to fruit 100 pounds of mushrooms a week? So if you're getting averages like me, my average right now, I'm averaging about 3 pounds, 2.8 to 3 pounds of first flush yield per block. So if you're getting something like that, then it becomes a simple math equation. You want 100 pounds. Let's just do 100. And you divide that by, let's just go a little low. And let's say 2.5 pounds for your first flush, because, if you're starting out and you're asking these questions like that, then you probably don't have a good baseline. And a good conservative baseline, you'll have positive surprises and very few negative surprises. So 2.5 pounds, so 100 divided by 2.5 is 40. And at that point, all you need to do is do 40 12-pound blocks a week, that kind of thing. And now it's a simple thing. You got to produce 40 a week. And if you want a little bit extra, just to make sure that you have enough for samples and that you have enough for any kind of losses that you have. Let's say that you have a batch that's contaminated, so you keep the last set for an extra flush so that it buffers some of your losses. Your yield goes down, but you still have that, and you do like 50 blocks a week or something like that, just to help buffer everything. What's up? Chip says, outstanding. More seat time on the bike. Remember, we do what we put our mind to and take action towards. It's much like starting your business. Keep it up, and I will make sure Instagram stays worth watching. Thank you. For sure. My bike really needs some work. Probably ought to just take it and get it worked on. See if there's a bike shop, repair shop that'll just do it for me. Because for me, it's going to be a big deal. But for someone who knows what they're doing, they'll probably just be able to throw it together and get it done. So maybe I'll just get it repaired and then start biking to work. I live 3 minutes away by car, and there's like one gigantic hill between me and work. But on the way to work, it's downhill, so I won't get all sweaty on the way to work. Just after. Do it. You have some great information. Thank you. Open source spreadsheet website for cultivators. I don't have anything like that. If you're in our Discord, I have put in like my experimental sheet, like where we're just taking our yield data and stuff. I'm not sure what spreadsheets you need, but in our Discord we share that kind of stuff quite a bit. Thank you, Cetus. I appreciate that. So we crushed some sugarcane, and we know it's winter, so to speak, in Florida. But man, what I started to think about when I started, I don't know, the chat jumped. When I started to think about the bagasse and the production quality for the 55G LC we were talking about. Oh, okay. There we go. That's what I was missing, man. I'm sorry. I was having a hard time parsing out that. That's going to be pretty cool. Let me know how that goes. And thank you, Magnetic. Maybe I will write that book. I'm working on a couple of articles right now for the website. Daniel said, I'd bench 80 kilograms 4 years ago and do countless pull-ups along with taking notice to my diet, though me and no one would take notice of my flapjack intake. Dark Waters is a movie, and there's a doc. A doc, though. So benching 80 kilograms. What is it? 80 kilograms would be about 160 pounds. Okay. I gotcha. Chad Amy. Hey, Andrew, how do you recommend a newbie should go about storing cultures they have isolated from MS? Multispore? I felt the need to keep taking agar transfers as a backup once a week. If you just want to store cultures, and they are cultures like oysters that can take cold weather, then you just do a culture slant and then store it in the fridge. And that's what I've done. Aislinn, can you turn the sound off on that? If you're doing something tropical, like something that can't take being refrigerated, then liquid culture is actually a really good way to store stuff. Put it in a syringe and store it. I've stored stuff in a liquid culture syringe for about a year. So, putting into words what I mean in real world times. Okay, I'll have to check out that movie then. Dark Waters. I'll check it out. Paul says, I made mead for the first time this year. Delicious. It's incredible, right? It's a happy drink. Unlike tequila, which is a fighting drink. Can you give some tips on when to know an LC is contaminated? I know without a microscope, you can't tell 100%. Well, you can't even tell really with a microscope 100%. The best way to do is test on agar. You just take up a little bit of liquid culture and squirt it on an agar plate, and it will contaminate like that. Let's see, if you don't have that, one of the ways to tell, like I can look at a liquid culture nowadays and basically tell, oh, thank you, Dan. I really appreciate that. To go towards mead brewing videos, there's only one person who I'll have teach me how to make liquid nectar. All right, Dan. All right. Your 5 pounds has been noticed, and it will happen now. we will make some more mead for sure. So we'll do, if it looks milky a lot of times, then that's bad. Like you should see some chunks, like a lot of times my liquid culture reminds me, like I'll swirl it and then look it up, and it reminds me of like Stellar Nurseries or something like that. Like you can see it when the water starts to clarify after about 10 days or so. That's a positive sign. Astronomy lasers are a really good way. Hold it up to the jar and run that astronomy laser through. If it doesn't penetrate and the water's murky, probably bacterial. When the astronomy laser hits the mycelium, it refracts, right? And it will start to glow, and it will highlight all the mycelium in there. And that is a really easy way to tell. And I used an astronomy laser for the first couple of years while I was figuring that out. One sec. Starting to get hiccups. I hope that helps. MCITP. I don't know how to pronounce that. And yeah, somebody here said, drop on the agar. That agar is the easiest way to tell. Meads lead to distilleries. Oh, I've had my time under the copper spiral plenty of times. Some species can't be bred, sterile cultures too. Yeah, I mean, from my understanding, there's quite a few fungal species that we haven't figured out yet. I think any species can be grown. It'll just depend on how well we figure that out. And Dan, again, thank you for the super chat. I was very excited. Somebody mentioned to me that I didn't have those working for some reason. I just got this is actually the first super chat I've gotten since I've turned those back on. I'm very appreciative of that. That did a dual thing for me. Number one, you gave me some financial incentive to start brewing mead again. And then secondly, you helped me test the infrastructure out and make sure it works. So thank you. Paul says, one question, I'll leave you alone. Oh, you're fine, Paul. That's what this is for. Flowhood, build or buy? If buy, any recommendations? If you want to buy one, so I would recommend, going through Ben. Like if you guys call our number, and Ben is building Flowhoods right now on consignment, or not consignment. Yeah, I mean, like if you call and you ask him about him, he'll give you a quote on one, he'll build one out, and then ship it out on a pallet for you. But if you want to get one from somewhere else, Eric Myers has some all-in-one units that are pretty good from what I hear. They're much cheaper than what you would be able to get from Ben. Hold on a second. Hey, Brody, turn the sound off on the game. Thank you. So Eric Myers has got some, I think that'll be about half the price is what we're selling, but they're the same filtration level at a reduced flow. That is not to say that they do not work. That is not to say that they do not work, because they actually work quite well. It is just that it is it's a ceiling unit. It is actually meant to be like in a drop ceiling with a bunch of them working together to make a clean room. So they're a little lower flow. That said, many people are producing really good work on them. So that's also a good way to go. And about half the price is what you would get from us. My current hood is too small and old in the market for a new one. So Ben's making some pretty good, comfortable ones. Like Eric Myers, I don't know when he's got some that are coming in. Really, that's just the big thing is I would just talk to Ben. Oh, another super chat. Thank you. Thank you, Lance. I appreciate that. Redheads rock. Oh, man. Fair enough. I'm a little partial to my blonde, but, let's see. Going back to the flow hood, that's the two suggestions I would make. You can also get them from like Fungi Perfecti. He gets his from, I think, is it American Flow Hoods or American Air? Or I can't remember what it's called. There's a lot of them out there. And there's not very many that I have a lot of experience with. I do have a Fungi Perfecti hood, and I have a Ben-built hood. And that's the only two hoods I've got right now. So I hope that helps. And Lance, again, thank you for the super chat, man. I really appreciate that. Daniel, assumptions are bad, I know, but the people in this chat so far are new unless they've seen your previous videos. Thank you, Andrew. You're very welcome, man. Yeah, I just always assume people are new. It makes life a lot easier. Sometimes I'll catch myself, and Ben catches me all the time, using, not acronyms, but, yeah, acronyms. And it's like, you need to just say the word. Don't say LC. Say liquid culture. It's not that much more difficult to say. So, do you have any thoughts on black agar? Any notable purposes or just a waste of carbon? So, I'll tell you, Hypnotoad. Nice Futurama reference, by the way. Black agar is something that I have done now a few times. I like the food coloring a lot better. I know there's a big thing out there where people are saying the food coloring is just a novel thing and it's not all that fun, but I think I am going to do a video on black agar versus the food coloring. Black agar, you cannot see through it, and I don't like that because I like to look up through the bottom of my dishes to look for contamination under the mycelium. On the surface of the agar, but under the mycelium, on the dish. And so One of the nice things about the food coloring is that most white rot funguses break that food coloring down. So I can color code all of my recipes with the food coloring. Um, Royal Green, for instance, is my MMP plus recipe, my master's mix. So it has a gram. It's a MMP plus, which stands for Master's Mix Peptone Plus. And what that comes down to is 500 milliliters water, 1 gram hardwood fuel pellets, 1 gram soybean hull pellets, um, a 20 grams of liquid culture premix, 20 grams of agar, 1 gram peptone, and that is put into a 1 needle full of food coloring gel. And that is then added to an Erlenmeyer flask, stuffed, put into a pressure cooker, and then cooked at 15 psi for 45 minutes. And that is my favorite recipe right now. I'm getting amazing growth. Doubling up on the agar has made it much easier to do needle biopsies. The agar catches in the needle and stores in the needle much easier than the softer agar does, so making it a little bit more hard and brittle. And the mycelium is not having a problem, getting through it. And so it's been working out really well for me. Peptone is just blowing my mind with how fast it speeds up culture growth. And then doubling up on the sugar was actually an accident, but it performed so well that I just have been using it. Now, here's why I like the liquid culture. I can color code each of my recipes. I can pick the Ziploc bag that has the recipe I want, go over to the flow hood. I pull out just 4 dishes because I'm back to storing in Ziploc bags because it makes being able to just grab 3 or 4 dishes much easier than having to open up the whole pack, keep them sterile, close them back up, do that whole thing. So if I want to trial out 3 or 4 different recipes, I can just grab a Ziploc bag of each and then go to the table. You transfer your culture onto that dish, and now it's growing out from that center point. As it's growing out, it's consuming the food coloring. So the dish becomes see-through without a green tinge or anything anywhere that the mycelium is growing. Now, let's say that I have some bacteria and the mycelium grows over that. When it grows over that, it does not eat the food coloring underneath that bacteria at the same rate that it is eating the food coloring everywhere else. When I take that dish and I look upside down, or I look through the bottom of that dish, there's a light on one side, and then I'm looking underneath. Oftentimes I'll go, oh, there's a green spot there, and I'll look a little closer, and sometimes you can see that circular bacterial colony or yeast colony right there. And man, it makes visual identification of things so much faster. So I'm finding that the food coloring is it serves a few different purposes, and I like that so much. The black agar, I can't see through it, and so it doesn't serve that same purpose, and it just seems like a waste of time to me. So I hope that helps, Hypnotoad. Again, love the name. Vance, what's up, Vance? I'm using your oat grain spawn method and was wondering how often to shake, when to shake, and when you think is ready to be used for inoculation. Maybe you can just do a video on this. Okay, I can do a video on that for sure. That'll be one of my quick tips. Maybe Monday I can video that. That'd probably be good because I've got some stuff growing in over the weekend. So it's never by the day. I don't have like a schedule for it, Vance. I just go in and I visually inspect my spawn bags. I'll spray it, like let's say I open up my bag and I spray liquid culture in through the top. I'll let it run down. I'll spray it all on that backside and let it run down, seal it up. Now, when I come in, as long as my liquid culture has grown, 3 grains over, so I've got 1, 2, 3 grains or whatever, that liquid culture sprayed down, and it's grown 3 grains in that direction, 3 grains in that direction, then it's created this very light colored. It's like liquid culture does not grow in very thick at first, typically. It usually grows in very light. I'll then, as long as it's grown 3 grains that way, 3 grains that way, and created a mass that way, I'll shake that up and disperse everything through the bag as easily as possible. Sometimes I'll even grab some of that chunk that's been grown in, and I'll shake everything else and then release, that chunk that I kind of held on to, and just make sure that it's got a good and even spread. And then my bags, they will grow in like 10 days a lot of times. And it'll be really, really light. And I use my bags when they're really, really light colored, like when you can just barely see mycelium all the way around the bag. But usually if you wait just a couple more days past that, that liquid culture is one of those things that grows in. It's really thin, really wispy, really thin, really wispy. And you can use it at that point. But then it'll grow in all of a sudden. Like you'll go to bed one day and you'll wake up the next and you'll come in and that whole bag will be solid white. You'll be like, oh my gosh, it's almost too late to use it. Like kind of like that. So, but I'll do a video on it. That hopefully that gives you my timeline and like what to look for, but I'll still do a video on it. That'd be a great quick tip. Chip says, you make a little coin now, and as a CEO, you never have enough time. The bike shop always is looking for and better yet, a long-term customer. That'd be a good idea. Ben and I used to bike all the time, and my goodness, when I was biking everywhere I went, I was lean. I was a lean machine, and I ate like crazy. I'd like to get back into that. Cetus, I think that you have got me. I'm going to get the whole family back on bikes. We'll just start biking around town all over the place. That'll be our weekend dates. We just ate gray dove oysters from sawdust spawn off of my 2014 sawdust spawn hidden back of fridge. Oh, 2014 the year. Nice. That actually sounds pretty nice. Has anyone been watching the latest offering of Star Trek? Commander Paul Stamets, the navigator of the spore drive, almost instantaneous travel along the interstellar mycelium. I have been watching that, Cameron. I actually really enjoy that show. I was really afraid that they were going to take that show because they were making it a big deal about, the lead character being a black female and having a big diversity of cast. And I was like, oh my gosh, please don't make this about, the race and then just have terrible acting. Like Star Trek has always been very diverse. That's the whole point. Like keep it diverse, but let's not like politicize the damn thing. Right. And I guess Star Trek's always been a little political, but my gosh, they show like, I like Star Trek Discovery. They did not over politicize it at all. It is beautiful. It is a great show, and I cannot recommend it highly enough. So, super chat time. Thank you, Paul. I really appreciate that, man. Lego Master, best wishes, and thank you all. Thank you for your selflessness to help us all out time and time again. You're very welcome, Paul. Thank you very much for the super chat. That means a lot to me, guys. I really appreciate all of that. Let's see. Randy Johnson, did you change your recommended recipe for LC? I've been using your 4-1 premix recipe. No, that's basically it. It's 4-1 or 5-1. I think right now I've been doing 5-1 because it's just easier. It's easier to buy a pound of malt and then 5 pounds of corn dextrose than it is to do 4 pounds of corn dextrose. But 1 and 5, 4 and 1, both of those are fine. I mean, 4-1 and 5-1 is just fine. Either one of those works really well. And that's what I use for my liquid culture premix. Matt says, love your show, bro. New flow hood and grid over the front to save the filter front is good. Yeah, I'm definitely thinking about that for the next production filter I get for the production lab. My lab, I'm not that worried about it because I'm really careful. And only I and one other person has unfettered access to that. And that's Jackie, our farm manager. But, beyond that, like that's the only two that have them. We're both very, very careful there. In the production lab, things are a little bit messier, Chad says, so if an LC is contaminated, fungal or bacterially, will any one single drip on agar show sign of contamination? Yeah, usually if your LC is contaminated, it's contaminated. There's usually not like it's hiding out in one little space or something. Shake it up if you're worried about it. Shake it up and then put a few drops. I usually put 3 or 4 drops. I think someone said drop it earlier, and that maybe it sounded like 1 drop. But yeah, do 3 or 4 drops. Your guidance is more than Southwest mushrooms on a personal basis. You've been more personal than them, though things should be taken into account on a relatable reasoning in your videos that are more relatable than theirs. Oh, well, thank you, Daniel. I appreciate that. I actually I have a mad respect for Southwest Fungi's like cinematography game. I don't know who he's got doing his videos for him. But man, some of his shots are so cinematic. Now I know they actually, I've seen them using a red dragon, and I just can't afford a red dragon. Like red dragons are so expensive, and there's just no way I can have that kind of video quality for a mushroom channel. I don't know how he managed it, but he managed it. So I appreciate that you think mine are more relatable. I do really appreciate his. Like he's got a good visual game going. It blows my mind. Matt found that 36 by 24 works better for bag inoculating, less contamination. Writable white tape over parafilm works good. Nice. So you found that 36 by 24 works better for bag inoculating, less contamination. 36 by 24 is the two ones that I'm using, actually, now I think about it. And I found that they've got plenty of room for, big-shouldered boys like myself, and just the speed of inoculation is so fast. So I really like that workflow. And you don't need two people working in front of a flow hood. Like, just literally someone inoculates, sets it to the sealer, seals it, and then just goes back to inoculating. The next person will take it off the sealer and keep going. And they can shake outside of the flow. Daniel says, sorry, my father has taught me and mother, and I've learned over years that it's the people that matter. Oh, I'm sorry, guys. Discord is popping back up. Let me mute that for another hour. All right. So, sorry, my father has taught me and my mother, and I've learned over the years that it's people that matter. Also, if you're able to learn, please do, because my grandparents, both sides, have to take into account our life. So, I'm getting very philosophical. Should have related the chat to people I know. Well, fair enough, Daniel. I appreciate that, man. I agree. People matter, and that's absolutely the way it works. And I was scared to start the YouTube channel. Like, I was like, my secrets, all my little precious secrets. Like, but I had a mentor too, and he helped me. In fact, he called me today, and I need to call him back. I was busy when he called, but anyways, I had a mentor, and I paid to take his class, and then after that, he never charged me again. He just taught me, and I've always appreciated that. And then beyond that, Ben came from the open source community. Like Ben has always been a big fan of the open source community, and we really started going into open source a lot. And the more I started looking into that, the more I really felt like it hit me philosophically that I don't need to just keep my secrets. The information should just flow. And I should just worry about making my nut on physical products, providing an actual value source for people, and then going from there. And so I really appreciate that. Like, it's kind of nice to have that feedback that you guys kind of get that I'm after people here, because these are my transmissions to you guys, right? And I'm teaching you my way. And in a way, it's selfish because I'm creating customers, like client customers, But on another point, like, I hope that I'm providing enough value to you guys that it is worth becoming a customer, right? Like, that's kind of the nice thing to go on about that. And it's really helped me connect with people. And this is my favorite part, is teaching people, talking to people. Like, it's up there with breeding, the mushrooms. Like, they've got the mushroom breeding that I really appreciate, but then that's my introvert side. And then my extrovert side is being able to just come in here and talk to you guys all day, on this or the Discord, and just really help people figure things out. It's really nice. Oh, John Grady, man, thanks. The spice must flow. Thank you for the super chat, John. And the spice must flow. I hope you're going to go watch Dune 2021 or whatever it is. I cannot wait for that movie. Is there any resources to look at to help understand what a healthy agar plate looks like and what the different contaminations look like? Thanks. Nature Man, if you're on Facebook, Agar of Asgard is a great place for a beautiful agar, and to see what the kinds of things that you can do with agar. It's a beautiful, beautiful place. Misty West has some just absolutely gorgeous shots. If you're wondering what that little circular cut that she does is, that's a biopsy tool. I think she does hers with leather punches. You can get dental, like discardable dental biopsies pieces and stuff like that you can do those little circle cutouts with. So, have you tried the Amber North Tech for vinegar water pasteurization of hardwood fuel pellets? I've had poor success at it. No, I have not. But that reminds me, I need to write this down, because Amber North messaged me recently, and I'm supposed to send her some cultures for her consult that she did with me. So bear with me while I write that down, because I forgot to send those out, and I do not want to piss them off. Cultures. I will email her after this as well. Anyways, I have not tried an aviator. The vinegar tech has never sounded like something that would work to me well, so I don't know anything about it. I do not like lime tech either. Like, I think lime tech is a trap. Everyone is always all about lime tech until they do it for a while, and then it crashes at some point. So, question from a noob: I am making light malt extract agar and have tried adding food coloring, but not having much success. It all seems to go to the same color. Is my weak British coloring? So if you're just using like the regular like water droplets, those are already it's not a concentrate. Those are already I can't remember the word. They're already it's had water added to it. You need a concentrate, something like a gel or a powder. That usually works best. It's not your weak British food coloring. It's just weak food coloring in general. And maybe it's alcohol instead of water. I think it's alcohol they're added to, but I can't remember the word all of a sudden. Why can I not think of that word? I'm starting to think floating mic on the surface of LC means trichoderma is likely present. What does your experience suggest, oyster hericium? I get mats all the time. Usually it's a weak stir bar, or I have forgotten to stir the cultures for 2 or 3 days. If you're seeing it spotted, hericium oftentimes will start these teeny tiny little circular colonies on the surface and then fruit off of them. And I've seen that a lot as well. So no, I don't think that floating mycelium is an indication of trichoderma. I will say molds tend to, like, if you want to test, a good passive way of testing a culture for mold infestation, not bacterial, but mold, is to let it settle, let some mycelium form on the top. If it turns green within 2 or 3 days, you know it's a mold, typically trichoderma. Speaking of, if anybody knows where a good trichoderma risi culture is, I am looking for trichoderma risi. So I would love it if somebody has one. I do not want trichoderma aggressivum. I already have that one. And if you don't know what I'm talking about, then don't worry about sending me one. So there's that. You love Southwest hair, don't lie. I do, man. Dude, his hair is gorgeous. I'm so jealous of his hair. It's beautiful. It just spills down. Crazy. Beautiful. Hypnotoad, what are your thoughts on mixing grain for grain spawn other than pellets in the bag? Being an oat man, have you tried mixing in millet best of both worlds or more work with hydration differences? I have, and the problem I had was that I was getting too many burst grains of one type and not another. And typically the millet would be what would burst. And I found with oats that if I didn't do a full good 2.5 hour cook, they weren't as clean as millet. So I just stick with oats because to me it's simplicity's sake. And now with the pellets in there, like, you can even add, like, if it's shiitake, add some coffee grounds. Spent coffee grounds in your spawn. A little shiitake LC. Shiitake loves coffee, and I don't know what it is, man. Just the way it goes. Let's see. Yeah, so the hydration difference. Sorry, I guess I didn't see that part. I read it, but I don't remember it. That I think is the big problem there. Dan at Fungal Fruit says, as far as I'm aware, oysters are tolerant of alkalinity, but not very good with acidity. Yeah, so I've done like coffee agars, and I did like the only thing that performed well in coffee agar was shiitake. All of my oysters stunted really badly. Chad, you're very welcome. John, again, the spice must flow. I actually read, what was it? Like, I'm in the beginning of the book. I think it's right here on Dune. Trying to find it. I won't spend long on this if I can't find it quickly. I probably won't be able to find it. Okay, here you go. I like this. Many have marked the speed with which Muad'Dib learned the necessities of Arrakis. The Bene Gesserit, of course, know the basis of this speed. For the others, we can say that Muad'Dib learned rapidly because his first training was in how to learn. And the first lesson of all was the basic trust that he could learn. It is shocking to find how many people do not believe they can learn, and how many more believe learning to be difficult. Muad'Dib knew that that every experience carries its lessons. From the Humanity of Muad'Dib by Princess Irulan. I love that line. I feel like that is one thing that separates a lot of mushroom people from other people is that I feel like mushroom people still believe they can. they can learn, and that learning is not difficult. All learning requires is for you to be present and do enough reps that you learn it. Like, that's it. Just show up and do the work and you learn. Like, it's so easy. And I feel like so many people discount themselves. So, John, thank you for that Dune reference. I'm glad you guys are geeking out with me on the Dune as well. People matter and numbers don't count. So, Lance, that's a good point. Like, I sometimes I get a little like, oh man, like 20,000 subscribers. What the fungus has got 50,000, or some, I don't know, some stuff like that. But that's not what counts. What counts is like, I have fun with this. You guys are helping support me. You guys are buying products. You guys are obviously interested in what I'm doing. And we're building a community that it's not just about numbers. It's a community of an exchange of information, of energy, and of material. And that exchange is a sacred thing. I freaking love it. I love it. So you're right. I'm with you on that one. Hypnotoad. The first movie was lacking the spice. Crap. First movie of what? Of Dune? Because I agree. The 1981 was not my favorite. It's a good one. I actually liked was it Dune 2000? I think that one was the one that I liked more. Even though the costumes were terrible and the CGI was, but they get pretty close to the story. Thank you, Andy. Another super chat. Aviator, thank you from Phoenix, Arizona. Well, thank you, Aviator. I really appreciate you guys, man. Thanks for hitting that, wearing that super chat button out. I really appreciate that, guys. That is going to, you've bought me quite a few Pal's cheeseburgers. It's one of my favorite lunches. It shouldn't be. And they've got cheese curds now. Oh, it's a good breakfast. So thank you for the cheese curds. He who controls the spice controls the universe. Damn straight. Did you guys know that Frank Herbert, according to Paul Stamets, I read it in Mycelium Running, Paul Stamets met Frank Herbert. Frank Herbert wrote Dune about mushrooms while on mushrooms, or at least based on a mushroom trip he took. So he wrote it about, like, the ecology of Dune is about the mushroom life cycle. The many of the things that he talks about are about mushroom trips and stuff like people experience in the book. And then he wrote that based on an experience he had while on mushrooms, is my understanding. So that's pretty neat to think about, right? Like, no wonder so many Dune people love mushrooms. There's so much I had to say, though. YouTube has deleted it when switching screens. Andrew, thank you. Oh, I'm sorry, man. Yeah, it does that. YouTube can be pretty picky on its chats. And Aviator, again, thank you. I hope I said thank you. I'm not sure if I said thank you for the super chat or not, but thank you very much. I really appreciate that. It's guys like you who defended from the prophets. Sorry, Chad. Okay. I'm going to have to see. It's guys like you who defended from the prophets, such as McKenna and Stamets. I feel like you are their pupils, bringing to life their visions. Imagine what future generations might do. I can see that, yeah. So that's been my one thing. There's a lot of people who get into this Paul Stamets hate. And while I don't like the use of bad scientific studies, and I don't like the kind of miracle cure stuff that he does, one thing that we can all agree on is Paul Stamets started a lot of this. Now, he's not the only one. Like you said, McKenna brought a lot of this stuff forward. Oh my gosh, I forgot his name. Met with Maria Sabina back in the 1950s. Oh my gosh, I can't believe I forgot his name. Oh my gosh, it's not gonna matter. I'm not gonna be able to remember it. If somebody can remember who that was, that did the journalist who did the story on Maria Sabina and kind of cracked open this stuff, please mention it in the comment or in the chat, please. But Beatrix Potter, who was a mycologist and wrote some of my favorite stories as a kid, like The Tale of Jeremy Fisher. That's always been one of my favorites. I still have a bunch of those little green books that my grandmother gave me. Oh gosh, there's so many. Gordon Wasson. That's it. I remember it now. Anyways, uh, what I was gonna say is that I feel like it is interesting how it's been building these little nodes on the network kind of hit. And Paul Stamets, I watched my 6 Ways Mushrooms Can Help Save the World, and then I bought mycelium running, and I had like $0 at the time. I just whatever money I made working a temporary part-time job, I spent on that, and then I started reading, and then I bought a kit, and then I started playing with lab work, and then I lost my job after getting a full-time job, and then went to college and was reading his books while taking microbiology. And the next thing you know, like I agree, like these guys started stuff, and what the Paul's impact on the community cannot be denied. People might not like the way he did it. People might not like some of the things he talks about. People may dislike his hippie nature. People like none of that stuff bothers me because I can at least recognize the good that he's done me, even if some other people don't like, or that there's some things that he may have done that people may consider bad. I don't know, but I like it. I like that thought a lot, and I hope that I hope I am furthering the mushroom way. Taking road man, but not actually interested in rap. Wait, what? Talking road man, but not actually interested in rap. I'm not sure what you're meaning there, Daniel. We were taught to grow in 1979. Our mentor told us, remember, this information is freely given. That's very cool, Aviator. I like that. Information, man. Information should flow. It's like spice. It must flow. Michael Robinson, back to the LC recipe, if I may. In radical mycology, he uses 1 tablespoon of LME and 1 tablespoon of dextrose for his LC. Did you try this before going to your 4:1 ratio, my friend? I have tried, I think that recipe. I've tried a bunch out, and I don't like it as much. I do add 1 gram of peptone to all of my liquid culture now, and I'm getting much better results, like much faster growth. So, LME and 1 tablespoon of dextrose works all well and good, like the 1-in-1, and that works, and I don't think that there's a problem with that at all. My first liquid sculpture. Yeah, try the recipe out. If you like it, you keep using it. What is the dental biopsy? I'm a dentist looking to leave dentistry to mushroom farm. Dental biopsy tool? It's like a little circular punch. That's what, I don't know why they call it that. It's just, as far as I understand, it's called a dental biopsy tool. DNTA, dental biopsy tool. Yeah, I'm seeing the set of 3 tissue punch dental surgical biopsy instrument, $12.99. I don't know why they call it that. Scientific Labwares has a set of different sizes for $30. Those are disposable. I do like this. Like on eBay, I'm seeing a set of 3 for $12.99. Looks like they're all steel, so they'd be multi-use. That would be very interesting. So yeah, there's a few. It's just a dental biopsy tool. I'm trying out your biopsy to LC technique right now, but forgot to put the stir bar in the LC. Oh, that's bad. I would just restart. You don't want to fight. You don't even want to fight it. Just restart it. What do you do with leftover blocks after you harvest the fruit? So right now, almost all of ours go into our landlord bought a trailer, a big, nice trailer, and he bought that, and that just sits outside the dock, and we just throw all of our old blocks in there, and then he takes that to his land, his farm, and uses it on his farm. The other side of that is I have started a superworm farm, and the superworms will eat fungal material like crazy. So we're about to start taking spent blocks and then adding them into this little farm and letting the larvae and the beetles and everything just live off that stuff. And even if it's a little less efficient than feeding them like vegetable matter and stuff, who cares if it's a little less efficient, because I have all the fungal biomass in the world that I would ever need. So, we're just working with that. We put them in gardens. We put them in just anywhere where we need good soil, and that's the way we go. Let's see. Ditto again. Oh, no, I lost the chat. R. Gordon Wasson. There we go. Yeah. I'm glad I remembered it, though. But yeah, thank you for putting that in there. Was that McKenna that went to Hawaii and heard voices. I don't know. I don't know much about McKenna. That was not the mycological school I came from, I guess we'll call it. I'm not entirely sure from that. Lance says, aloha from Makawe. Oh my gosh, I'm going to butcher that. Makawe? I don't know. So I've got 2 employees that work for me, and they're Cherokee, and they were telling me, I think they have some, I don't know if they were saying that they were like Tongans or if they were Hawaiians, but they were telling me that they've got some people that come, I think it was from Hawaii, but I don't remember which island, that come to like their stomps and stuff, or their powwows rather, and come and dance and do different dances and stuff with them, and that sounds pretty neat. I'm gonna have to go to that when things open back up. Like I think the Cherokee Nation has shut down all activity, like the powwows. When they start that back up, I'm definitely going to go to some. It'd be very interesting. Maybe wearing like my formal kilt and stuff to that. That'd be kind of neat. I've just shown my grandparents some factual videos and have been showing them your channel for a while now. Things like you say have been picking up. England isn't a straight reaction though from the movies and documentaries I've been watching for years. Are really good. I'll message you and move. No worries, Daniels. This is fine, man. Lance says, I just went to the dentist office today. Stay in dental science. Oh, yeah, that's fair, man. It's hard to exchange that kind of labor, that kind of a man-hour to dollar value, isn't it? How much Peptone for 500 milliliters LC? Dillinger, when I make 500 milliliters LC, I just use 1 gram Peptone. Check out Hamilton's Pharmacopoeia. There's 2 mushroom episodes, among others, with a scientific perspective. Good stuff. Okay, I'll check that out. I've watched one of his things before. The kid who does the interviews kind of freaks me out. He's a little weird, but I guess that's Hamilton. I don't know. But some of the people I've seen him interview have been pretty neat. Terrence McKenna lived in Hawaii and started a garden that has different plants that are exotic, you could say. I bet he did. Knowing who Terrence McKenna is. Oh, my goodness. Well, guys, I think I'm going to jump off here because I've gotten down to the bottom of the chat, and we've had a very good evening, and I think it's time for me to relax. It's almost 9 o'clock, and my kids are going to be going to bed in about 15 minutes. So I'm going to tell them good night and snuggle with them a little bit. Or if they're too busy on electronics, maybe I'm going to jump up on I'll jump on Apex and play or something. So if anybody's into playing Apex, my gamer tag is Myco Miner, MycoMiner. I'll be happy to play with any mushroom loving people. Just send me a message. But yeah, guys, with that, I love powwows. I was born in Fairbanks, Alaska. Keep ticking. Yeah, heck yeah, Lance, that's awesome. We need to see, like, when they have a powwow up here, and Jackie, my farm manager, he dances. When he dances, maybe we'll see if he'll allow us to announce it and video it and stuff. Maybe we can put some up on the channel. When I do the Highland Games, I'd like to put some of that on the channel too. So yeah, we'll get it, guys. Thank you all very much. Have a very good evening. As always, y'all keep spawning culture. And by the way, I don't know what was up with that last video where I was all like, keep spawning culture. I was like stroking out or something. So anyways, guys, thanks, Farmer Mac. Have a good evening, fellas and ladies.